So it took about a little over a decade to develop the first generation of these inhibitors that span 3 different classes. And while this made a huge impact on the epidemic and suppressed the virus replication and helped basically halt the progression to AIDS, it wasn't all great. And that's shown here is this is what an average patient was now dealt with. And so now, in 1996, they all had a new lease on life. And the point is these drugs had to be taken at different hours, some with food, some without, and you basically had to have a little booklet to track your medications. And this really made it difficult for patients to live the life that they now had. And so we and others spent quite a bit of effort in trying to simplify regimens. And it took a decade, but we released the first single tablet regimen, shown here, a tripla, which is a 1 pill once a day. And this really made an impact in the efficacy of these therapies. They improved compliance and improved or reduced resistance that emerged. This was really just the second wave in improvement of therapies. But we don't end here. There are some toxicities associated with many of these single tablet regimens. And also, as we've heard in the first talk today, it's not a cure. So basically, Gilead started in 2006 and continues on to do 2 major things. One is to continue to improve these single tablet regimens. The second is to identify novel antiretroviral targets to combat emerging resistance that still occurs that these current STRs cannot handle. And thirdly, there's a huge effort, and this is very, very much a global effort, to develop effective activate and eliminate curative strategies. And so all of these are ongoing, and I'll just be talking about this first point. So the approach that we took, we went after many targets, and I'll just jump to the punchline. Many of them failed, and I'm going to illustrate through the course of this talk what goes into making a clinical drug in a novel mechanism. And I'm going to illustrate one that actually panned out, but it wasn't easy, and hopefully you'll understand what went into it. The target we went after in this case was HIV capsid, and this is a very non-traditional target because it has no enzymatic functions. It's purely structural. It's initially expressed as part of a precursor polyprotein, either GAG or GAG-POL. It's cleaved by the viral protease to release mature capsid. And this single protein, in the context of the constraints of the virion particle at high concentrations and in the presence of the nucleic acids, spontaneously assembles into this conical core capsid. 1,500 monomers go into this, and they assemble into 2 basic structures. There's 250 capsid hexamers comprised of basically 6 capsid monomers and exactly 12 capsid pentamers that help form these defects to close this cone. This structure is essential for viral replication because mutations in capsid that impair the proper formation of this structure are defective for infectivity and most notably for reverse transcription. So we rationalized if we were to screen for compounds that bind capsid and specifically assembly, we could potentially impact viral replication at either the capsid assembly stage or additional capsid functions that had not yet been described. So the way we started was to take an assay that was very, very simple, and it's based on the notion that soluble purified capsid protein in the presence of high salt will spontaneously assemble into these capsid helical tubes. And you can think of these as basically unclosed capsid shells because they polymerize and they're comprised exclusively of capsid hexamers. The problem with this type of an assay, which we wanted to develop in a high throughput fashion, is it requires a lot of protein. And luckily we had some people at Gilead who had just come from the actin and tubulin fields. And if you are aware of that, these proteins are frequently purified via a repeated polymerization and depolymerization. And so they developed a procedure based on ammonium sulfate, and they cycled through basically forming these aggregates of capsid. They would pellet those down and they would recycle this. And in basically one day, they could take 6 liters of culture and isolate 10 grams of purified protein, which was automatically functional based on the way it was purified. So we used this protein to develop a high throughput screening approach, specifically looking at small molecules that could impact capsid assembly. And we modified the rate of assembly in this turbidity assay, which basically scatters light, and you can follow the assembly on a spectrophotometer. And we modified the rate of this by basically regulating the amount of salt that's present. In one scenario, we screened for inhibitors, so things that slowed the rate and extent of in vitro capsid assembly. We identified a ton of different chemical chemotypes, and they all universally bound to a single site, an induced fit pocket at the base of the capsid N-terminal domain. In cells, these inhibitors, in some cases, after optimizing them, also prevented capsid assembly. So rather than seeing that full conical core, you're only seeing basically a nucleic acid blob shown here. So there's no actual structure here. These had an antiviral effect in HIV replication assays, but in numerous attempts over multiple years in trying to optimize these, we were never able to get very good potency. So we were pushing 1 micromolar or below, and they often cross-reacted with HIV reverse transcriptase. The second approach we took was to basically slow down the assembly reaction and now look for things that increase the rate and extent of capsid assembly. And you can see, rather than these nice long tubes, we get tons of these small little malformed little capsid structures. And these universally bound a second independent site, which was both highly conserved and preformed rather than an induced fit. And so this enabled us to use structure-guided drug development because it was much easier to get a co-crystal structure, and the chemists could look at these and decide where they could modify the compound to improve its potency. So again, in cells, these accelerate now capsid assembly, but in uncoordinated fashion, and you get these really weird shaped capsids. And even though you have a capsid, it needs to have the proper shape, otherwise it just falls apart at the wrong rate and you do not support reverse transcription. So this also induced an antiviral effect. And so the question became, we now have 2 classes with multiple chemotypes. Which do we focus on? Because we don't have endless chemists on any given project. So one of the things that was really helpful in forming these was we did a number of different binding characteristics of different capsid forms. So we looked at capsid monomers and capsid N-terminal domain, and all the site 1 inhibitors bound exclusively to capsid monomer. We also looked at oligomeric forms, so capsid hexamers and capsid pentamers that form the conical structure. And the site 2 capsid binders bound very well to the monomer, but they bound tenfold better to both the hexamer and the pentamer. They also bound immature precursor forms, GAG, by SPR. So to us, this suggested that this was highly more advantageous than the site 1 compounds because you have the prospect of binding and inhibiting all different stages of capsid function. So I just want to focus your attention on, this is a capsid hexamer, and you can see one of the neighboring monomers is highlighted in a lighter green, and this is one of our advanced inhibitors, and where it binds, this is the site 2 pocket. And what you can see is that it binds at the interface of 2 neighboring monomers, and now it makes perfect sense why you're accelerating misassembly, because in essence, it's sort of the glue that holds things together. And so what I'll show you is that these inhibitors bind at this site, and they can alter both capsid assembly and disassembly. And what is not shown here is this site is highly conserved because it normally binds host nuclear import and trafficking factors, and so the compound has the potential to directly compete with these essential host factors. And just one basic overview of a very complicated, this actually makes me depressed because I spent almost a decade trying to decipher this, and it's now in one simple slide. So the message I want to convey is, although we started with looking at capsid assembly, actually capsid is a multi-stage mechanism of action. It inhibits both late processes, so capsid assembly. In data I don't have time to go in, the inhibitors actually bind to the precursor polyproteins, and by a mechanism we don't fully understand it yet, it triggers the destabilization of the protein, and you now get reduced production of virus particles. So you have a 2-for-1 here at the late stage. You make less particles, and the particles you make get basically screwed up capsids, which are now no longer infectious. But what was very interesting was, capsid plays a role in controlled uncoding to support reverse transcription, and I told you it interacts with host nuclear import factors. So the compound actually also slows down the complete uncoding of capsid from this pre-integration complex and directly competes with these factors. And what this does in an imaging analysis shown here in primary CD4 T cells, the viral DNA is denoted by these red dots and capsid is highlighted in green. And you can see in a mock-infected cell, you have viral DNA 12 hours post-infected exclusively in the nucleus, and capsid is either outside or inside of the nucleus. But when you add the capsid inhibitor, you don't affect reverse transcription at these levels, but you do prevent it from getting into the nucleus. And you get now capsid aggregating in the cytoplasm. It's very tempting, but please don't think that. It looks like an intact capsid, but that's not what we're seeing here. And basically, you're getting accumulation of the viral DNA in the cytoplasm. It can no longer get in and integrate. So I guess the important thing from all of this is that it actually leads to an antiviral effect, and this is the clinical compound that we ultimately developed, and in comparison to current antiretrovirals, this is an MT4 T cell acute infection assay with a laboratory 3B strain, and 6207, our clinical compound, is significantly more potent. This is 100 picomolar, and on a protein-adjusted EC95 basis, it's more than tenfold more potent than all current antiretrovirals. Importantly, we also find that it's equally, if not more, potent against a broad panel of HIV-1 subtypes shown here. So this is the MT4 data, and these are PBMCs infected with the different HIV-1 subtypes, and you can see We have a mean EC50 of 50 picomolar. There's no major loss against any one strain that we've tested thus far, and that's consistent with the high conservation of this protein. In terms of drug development, it's important to make sure that you're not cross-reacting with some of the other resistance mutations that emerge in other current targets. And so the take-home message here is we looked at a panel of HIV variants that are resistant to different classes, either maturation inhibitors, protease inhibitors, or two different flavors of the reverse transcriptase inhibitors, or the integrase inhibitors. And you can see that the controls, they're highly resistant within a given class, but there's no fold resistance against our compound. Also, when tested in pairwise combination, 6207 is highly synergistic with each of these class members. And also, when you look at the cytotoxic potential in various in vitro cell lines and in primary human cells, we basically see no observable cytotoxicity up to the highest concentration tested here, suggesting that it has low cytotoxic potential. If you're going to make at least once a day drug or better, it needs to be rock solid stable. And so this data is shown here in 4 different non-clinical species. There's almost very little to no turnover at all of the parental compound. And in human, it's actually the most stable of all. From this data, it's predicted that 6207 will have low hepatic metabolic clearance in humans. So I want to bring it back to where I started, making one pill once a day. And this is, you could look at this and say, okay, great, make a new single tablet regimen, put this in there, and people who have resistance, this is perfect, and it's ultra potent, maybe you can make a smaller pill. And we will do this. But let's take a look from the patient perspective. That's one pill once a day. But this is what it looks like over the course of a year. And, you know, I don't know if people can really appreciate this from someone with a chronic disease, is any one of these tablets, you forget to take it. You were on vacation. You're around people, you don't feel comfortable taking your medication. And there's any number of reasons. And when you don't take your medication or you forget, then you have the chance of developing resistance and you've now failed and you have less options. Also, every time you take one of these medicines for a subset of population, it's a constant reminder that you have a disease. And that actually just, its psychological burden cannot be overstated. And so what we've heard from a number of patients is that they have a high interest in taking their medication less frequently. So either monthly, weekly, or it'd be great if it was once a year or quarterly. But the problem is trying to deliver this whole bucket of drug in a controlled fashion in a single administration. To make a long-acting antiretroviral, you really need 3 things working in concert. You need super high potency, you need basically low to non-existent clearance, this is the exit rate, and you need low solubility, this is the input rate from a drug depot. So the amount of drug that gets released from that drug depot site at any given time. And these 3 parameters together dictates the dose that would need to be administered over a given interval. So luckily, this compound has very low aqueous solubility, and it has very moderate binding to human plasma proteins. And these properties make it well suited for an extended release parenteral formulation. So we asked whether this actually would make a long-acting product, and we played around with different formulations and different dosings, different concentrations, and I'm just showing you one example of this extended release, in this case, in rats. So a single administration of 6207 is showing prolonged exposure well above the protein-adjusted EC95 target out past 6 months. So this suggested the potential for a monthly or greater dosing interval in humans. So this is unpublished data, but I think it's soon to be published, so I'm going to share it, and they're okay with that. So normally we can't. Before we initiated clinical testing, we did a proof of concept efficacy study in humanized mice that were infected with HIV-1, and we compared either obviously placebo, once daily triple therapy shown here in green, or every other week single injections of either a long-acting rilpivirine as a control comparator study to long-acting GSCA1, which is a close analog of our clinical compound. In data that's not shown here, the rilpivirine and the capsid compound had overlapping exposures, and we actually optimized this regimen for this study specifically to achieve that. Over the course of this 12-week regimen, we saw very little change in body weight, and there were no deaths associated in any of these groups. And you can see in comparison to the triple therapy, which rapidly suppressed over the course of the therapy, the long-acting rilpivirine had transient suppression and then rapidly developed resistance. And this is not unexpected of a monotherapy, which is why we developed these single tablet regimens. What was actually really interesting was that at the same exposure levels, the capsid tool compound lasted quite a bit longer, and only one mouse failed at week 12 of the study, and it developed resistance that mapped to the capsid binding site. So this gives us basically, this suggests that we have a good chance of this working in the clinic. So although the efficacy studies are currently ongoing, and that will be released, I think, next month at IAS, all I can really share with you at this point is the first-in-human safety, tolerability, and PK studies in healthy human subjects. And we did basically 4 different cohorts, and the data are shown here. That it's still blinded, but the different treatments were well tolerated, and there were no major adverse events. And at the higher doses, approaching 300, 450, we had therapeutic concentrations for 3 months. So this just brings us back to sort of the different ways of HIV treatment. So first, this was a very important step. This saved a ton of lives and continues to save a ton of lives. But basically, patients are looking at the clock. It's hard for them to live their lives. This transition to the single tablet regimens, which is basically a daily reminder, is very important and continues to be important. We're now transitioning, at least with a subset of patients, to the prospect of a seasonal, long-acting injectable. And so I just want to finish with one thing to illustrate to people who maybe are thinking about industry or have thoughts about what it's like at industry. This is one example of a drug development program. So many failed along the way. This is one that succeeded. We started in 2006, and this actually is where I joined. I had never worked on HIV, and it saddens me to see that it takes 10 years. But, I feel very proud of all the people that worked on this, and there's hundreds, if not thousands, that touched this project along the way. And we had many ups and downs, covering many different facets to get to this clinical candidate. And I just want to end from that notion, and I cannot thank enough people, because it would be a size 1 font. And we've had slides like that, and it just doesn't do any justice. But I want to recognize the team and some of the key players in the data that I illustrated here, as well as many of our collaborators and also all the participants for our clinical studies. So thank you, and I'll take any questions.
Thank you very much. Any questions? Phil.
So really fun to see behind the curtain there. I had a question about your cell-free system when you're doing the capsid assembly. How did you control for cyclophilin concentration, cyclophilin A? Did you have a source of that, or because that's in the cells, you know, it's incorporated into the capsid?
So you're talking about the in vitro capsid assembly assay?
Yeah.
Yeah, actually you don't need cyclophilin at all, and we've looked at adding recombinant cyclophilin into the reaction, and if anything it actually dampens it, but very slightly. But we've also looked at the mechanism and whether it's codependent on factors such as this, and in all cases it does not appear to be. So if we use a cyclophilin mutant, capsid mutant, there's no change in efficacy at all.
I have a question. Given the higher potency of your capsid inhibitor, do you have any idea whether it better suppresses residual replication and you know, or you have less residual replication and maybe a faster way to eradication, you know, or is that nothing that you guys looked at?
So that's a great question. We'll find out with the current trials that are ongoing. We've looked hard at this. We don't currently see how this will necessarily lead to the eradication effort. But we don't know yet because, you know, one thing that I pointed out is we are accumulating the viral DNA in the cytoplasm that may contribute to innate immune responses, but we also don't know capsid is accumulating there. We don't know if that may shield it. There's just too many unknowns. We will look at this, obviously, but we don't have any evidence of that.
Any more questions? Yes.
So lots of other viruses have structures solved for capsid as a monomer and also as part of the whole virus. Do you think focusing on conserved parts of capsids for other viruses might be a nice point to attack?
Absolutely. I mean, that's the thing is, you know, I'm sure there's some exceptions, but I think a lot of viruses use capsid as not only their protector, but as sort of their vehicle to get around, transport along microtubules, to find their way. I think it's an excellent target. But you said it. You need a conserved site that is druggable. And I'm sure people are doing this.